The last 12 lectures that we discussed in this course, namely lectures 1 to lecture 12, we primarily focused about regulation of messenger RNA synthesis, how RNA polymerase II transcription is regulated. We studied in depth the various general transcription factors that are involved in polymerase II transcription. We also studied how chromatin plays a very important role in the regulation of RNA polymerase II expression, gene expression, and we also studied how post-translational modifications of histones and as well as DNA methylation plays a very important role in the regulation of polymerase II transcription. And we also studied what are the various RNA processing events that take place, namely mRNA capping, mRNA splicing and polyrylation, and how all these activities are integrated. So, the first 12 lectures we confined ourselves to the regulation of transcription by RNA polymerase II, how messenger RNA synthesis inside the cells is regulated and how RNA polymerase II is able to synthesize messenger RNAs at different levels. How different protein coding genes are transcribed at different levels. Some are housekeeping genes, some of them code for proteins which are all the time synthesized, which are constitutively expressed. There are some genes which are induced all the time, and we studied how transcription factors through their DNA binding domains go and bind to specific upstream sequences, and through their transcription activation domains interact with chromatin modifying proteins or histone modifying proteins, and as a result enhance the recruitment of RNA polymerase to the promoter, and that is how the rate of transcription is initiated. So, the first 12 lectures we confined ourselves to only RNA polymerase II and protein coding genes, regulation of protein coding genes. So, in this class today we are going to talk about the regulation of ribosomal RNA synthesis, how ribosomal DNA is transcribed, or how RNA polymerase I activity is regulated inside the cells. So, eukaryotic gene regulation, synthesis of ribosomal RNA by RNA polymerase I, how RNA polymerase I makes ribosomal RNA and how is its activity regulated inside the eukaryotic cells is going to be the crux of today's lecture. Now, although we spent about 12 hours discussing the regulation of messenger RNA synthesis and regulation of protein coding genes, remember messenger RNA accounts for a very small fraction of the total RNA. If you ask me what are the most abundant RNA inside the cells, if you isolate RNA and run it on a gel, you actually see 3 major bands, 28S RNA, 18S RNA and 5.8S RNA. These are nothing but ribosomal RNAs. In fact, almost 50% of the nascent RNA which is synthesized in the cell is accounted by for the transcription of ribosomal RNA genes. So, rRNA transcription or the rDNA transcription accounts for more than 50% of the total RNA inside the cells. So, messenger RNA is only a very small fraction. The ribosomal RNA followed by tRNA and mRNA accounts only a very small fraction of the total RNA. So, it is important for us to understand how does the cell synthesize such large amounts of ribosomal RNA. Okay. So, regulation of RNA polymerase I transcription also as important and as vital as study and understanding the regulation of RNA polymerase II. And in the next class we will also study the regulation of RNA polymerase III, how tRNAs are synthesized, how Pol3 makes transfer RNA inside the cells. Now, transcription of ribosomal genes occurs inside the nucleus, nucleolus within the nucleus, and nucleolus is actually the site of ribosomal biogenesis. So, in order to make this huge amounts of ribosomal RNA, because they are the proteins, because they constitute the ribosomes, and the cell has actually identified a very specific region inside the nucleus which is the nucleolus, and it is inside the nucleolus that the ribosomal DNA transcription takes place. Ribosomal RNA is synthesized, processed, and ribosomes are assembled inside the nucleus and they come out. So, there are several hundred copies of ribosomal RNA genes inside a eukaryotic cell nucleus, because if you have to make such huge amounts of ribosomal RNA, you require multiple copies of the ribosomal genes. Therefore, there are many such several hundreds of such ribosomal genes inside the eukaryotic cell nucleus, and all these genes are arranged in clusters as tandem head to tail repeats, and what constitutes what is called as a nucleolar organizing regions. So, what I am trying to say from this slide is that ribosomal DNA transcription constitutes the majority of major transcription activity inside the cells. Ribosomal RNA constitutes more than 50% of the total RNA inside the cells. The ribosomal DNA transcription takes place inside the nucleolus of nucleus. Not only ribosomal RNA is synthesized, it is processed, and ribosomal assembly also takes place inside this nucleolus. And to make these huge amounts of ribosomal RNA, organisms have evolved, eukaryotes have evolved to have multiple copies of these ribosomal DNA genes, and therefore many of these rDNA genes, many hundreds of copies of these rDNA genes are present in a head to tail fashion, in a tandem fashion inside the in the genome, and they are actually present inside the nucleolus. So, nucleolus is the site of ribosomal DNA transcription and ribosomal RNA synthesis. And it is estimated that in the human HeLa cells, for example, about 40,000 ribosomal subunits leave the nucleoli per minute. So, just imagine the kind of transcription activity and ribosomal assembly that is taking place inside our cells. 40,000 ribosomal subunits leave every minute from the nucleolus, because unless they come out and they are the protein synthesizing machines, and unless they are present in large amounts, proteins cannot be made. So, in order to synthesize proteins in large amounts, especially in active growing cells, the ribosomal transcription is very essential. So, the transcription regulation of ribosomal DNA takes place depending upon the physiological status of cells. If a cell is differentiated, if it does not actively divide, the ribosomal transcription is very low, but in case of actively growing cells and actively dividing cells, the ribosomal DNA transcription is very high, because proteins have to be made in large amounts. Now, the nucleolar RNA polymerase I activity inside the eukaryotic cells is harmonized with that of nucleoplasmic, that is extranucleolar activities of RNA polymerase III and RNA polymerase II. This is also very important point. Now, if you want to make functional ribosomes, it is not just RNA polymerase I which is involved. It also involves RNA polymerase II and RNA polymerase III, because the ribosomes consist of not only 28S, 18S and 5.8S RNA, which are actually made from the ribosomal DNA. It also contains 5S RNA, which is made by RNA polymerase II so RNA polymerase III, and at least 70 different ribosomal proteins have to now combine with these ribosomal RNAs, and all these proteins have to be synthesized from messenger RNA. So, if you want to have a functional ribosome, you require all the 3 RNA polymerases have to work together. So, to make functional ribosomes, all the 3 RNA polymerases have to work together. RNA polymerase I makes 28S, 18S and 5.S rRNA. The messenger RNAs for at least 70 different ribosomal proteins are made by RNA polymerase II, and the 5S rRNA is actually made by RNA polymerase III. So, all these 3 RNA polymerases have to work together in order for you to get a functional ribosome. Now, let us now stick today about the synthesis of 28S, 18S and 5.S rRNA from the ribosomal DNA by RNA polymerase I. How does the synthesis take place and how does the regulation take place? As I told you, in order to synthesize these huge amounts of ribosomes which the cell requires to make proteins, the ribosomal DNA has to be present in multiple copies, and as I mentioned earlier, several hundred copies of ribosomal RNA genes are arranged in clusters as tandem head to tail repeats in the genome, and they are all present inside the nucleolus. And what I have shown in this cartoon is how these rDNA clusters are arranged, and you can see this green and blue together represents one rDNA repeat, which is about 43 kilobases long, and such rDNA repeats are arranged in a head to tail fashion in the genome. Several hundred copies of such rDNA units are present at a given stretch in the inside the genome, and they are all transcribed within the nucleolus. So, each rDNA repeat is approximately about 43 kilobases in the length, and it contains 2 important regions. One is called as the intergenic spacer, and another is called a region which actually codes for the ribosomal RNA genes, a non-coding region and a coding region. The blue 13 kb region actually codes for the ribosomal genes, and the green one, which is about 30 kb, is actually called as a intergenic spacer or IGS, which does not code for anything. Now, let us now see how exactly this ribosomal DNA unit is there and how exactly the regulation or synthesis take place, and this cartoon actually tells you the coding region, the 13 kb actually contains genes that code for 18S, 5.8S and 20S ribosomal RNA, and this is the intergenic spacer, and this is the promoter from which the transcription start site, the arrow indicates the transcription start site, and flanking the transcription start site you have the promoter, and the promoter is extends both in the transcribed region, the downstream of transcription start site as well as the upstream of transcription start site, and extends usually from -156 to +18 of the transcription start site. So, the ribosomal RNA transcription or the rDNA promoter extends about minus 156 to plus 18, and this is a key regulatory region for ribosomal DNA transcription. This is where all the action is, and this is where we are going to discuss what kind of transcription factors actually are present or interact with between the minus 156 region to plus 18 region. And within this region there are two important cis-acting elements, just like we have the Tata box, the near upstream elements, far upstream elements, and transcription factor binding sites in the case of the RNA polymerase II promoter, which we have discussed extensively. In the case of RNA polymerase I promoter, you have two important cis-acting elements. One is called as the core region, another is called as the upstream control region. The upstream control region extends from minus 156 to minus 107, whereas the core region extends from minus 45 to minus 18. So this core region is similar to the initiator or the Tata box region of the RNA polymerase II promoter, whereas the upstream control region is similar to the enhancer or the upstream binding sequences of the RNA polymerase II promoter. So just as you have a basal transcription factors which are assembled near the Tata box or initiator in the case of RNA polymerase II promoter, similarly a number of basal transcription factors in RNA polymerase I, there it was RNA polymerase II, here it is RNA polymerase I, along with certain basal transcription factors assembled near the core promoter, whereas to enhance the rate of transcription initiation by this pre-initiation complex assembled near the core region, a number of protein factors bind to the upstream control element and enhance the rate of recruitment of RNA polymerase I, therefore the rate of transcription initiation can be enhanced in the RNA polymerase I promoter. So Pol-1 initiates transcription of the human RDA promoter, which is designated as P here, which contains a core promoter element extending from minus 45 to plus 18, and it also contains an upstream control element or UCE, which extends from minus 156 to minus 107 region. There are several transcription termination elements which are present at the 3' end of the transcribed region of rRNA, which are designated in the red color designated as T here, and they are also present immediately upstream of the RNA gene transcription site, which are designated as T0 between the spacer and the gene promoters. So there are two different kinds of transcription termination elements, one immediately after the transcribed region and one just upstream of the promoter region. They are designated as T as well as T0. Now, as I said, this is the coding region or the transcribed region of the RNA polymerase II rDNA unit, which contains genes that code for the 18S rRNA, 5'S rRNA, and 28S rRNA. And all these three are recognized as a precursor molecule, which is called as the 45S pre-ribosomal RNA, and this 45S pre-ribosomal RNA is then cleaved to get the three individual rRNAs, namely the 18S rRNA, 5'S rRNA, and the 28S rRNA. So a single precursor rRNA is synthesized from the ribosomal DNA unit by RNA polymerase I, and this rRNA is post-transcriptionally cleaved to the three different ribosomal RNA molecules, namely 18S rRNA, 5'S rRNA, and 28S rRNA. So the primary transcript 45S pre-RNA synthesized by mammalian RNA polymerase I is processed or post-transcriptionally cleaved to mature 18S, 5'S, and 20S rRNAs, and all these three RNAs together with the 5S rRNA transcribed by RNA polymerase III constitute the RNA components of the ribosome. So a ribosome is a ribonucleoprotein particle, it contains proteins, it contains RNAs, and of the 4 different RNA molecules which are present in the ribosome, 3 of them, namely the 18S, 5'S, 20S are made by polymerase I, they come from the ribosomal DNA, and the 5S RNA is actually RNA is transcribed by RNA polymerase III, and together they constitute the RNA component of the protein. And in addition, messenger RNAs corresponding to at least 40 different ribosomal proteins have to be transcribed by RNA polymerase II, translated into proteins, and these proteins then interact with these RNAs and then to get a functional ribosome particle. So the 28S and 5.8 rRNAs associate with the large ribosomal subunit, which is what is shown in this cartoon. The 5.8S and the 28S, 5.8S and 5.8 and 5S are associated with the large subunit of the ribosomal particle, namely 60S, while the 18S rRNA associates with the small subunit or the 40S subunit of the ribosomal particle. So together 60S and 40S together form the functional eukaryotic ribosome. So just as the protein coding genes are transcribed by RNA polymerase II, the rDNA is ribosomal DNA is transcribed by RNA polymerase I, which we all know very well. The yeast RNA polymerase I, we knew from our previous 12 lectures that the eukaryotic RNA polymerase II is a multi-protein complex consists of a number of subunits. The same way, the polymerase I also consists of a number of subunits, at least 14 of them are there in the case of yeast cells. And there are mammalian homologs for all these 14 which have been first identified in the yeast cells, except for one of the yeast homologs is missing in the humans, namely the RPA protein. What I am trying to say is that RNA, just like RNA polymerase II, RNA polymerase I also is a multi-subunit complex. It is a huge protein. And as observed in the case of RNA polymerase II, Pol-1 transcription also requires Pol-1 specific transcription factors for promoter recognition. So the mechanisms are more or less similar, but the players are different. So just like the RNA polymerase II require the basal general transcription factors like TF2D, TF2E, A, B, E, D and so on and so forth, the same way the RNA polymerase I also requires certain general transcription factors for accurate initiation of transcription from the rDNA promoter. These general transcription factors which are required for RNA polymerase I are designated as TF1, whereas in the case of RNA polymerase II they were designated as TF2, TF2A, 2D, 2D and so on case, where in the case of polymerase I they are designated as TF1, A, E and so on and so forth. Similarly, in the case of RNA polymerase III, they are designated as TF3, A, B, C or TF3, 1, 2, 3 like that. Now, what are the cis-acting elements of RNA polymerase I promoter? As I mentioned, there are two major cis-acting elements. One is called as a core region, which extends from -45 to +18, and the transcription start site is included within this. And you also have what is called as upstream control region, which extends from -156 to -107, and in between you have what is called as the spacer region. Now the core promoter is GC rich except for a short stretch of AT rich region around the transcription start site, which is the equivalent to the initiator of the RNA polymerase II promoter, and the core promoter is sufficient for basal levels of transcription, but the efficiency of transcription can be further enhanced if the upstream control element is present upstream, which is also rich in the GC residues. Just as people have mapped the important nucleotides, important regions required for Pol-2 transcription, in the case of Pol-1 transcription also, if you make mutations within the core region or within the upstream control region, there is a dramatic decrease in the transcription efficiency, clearly indicating that these two regions play a very important role for transcription initiation by RNA polymerase I. Now, so two major cis-acting elements are present in the RNA polymerase I promoter, the core region as well as the UC or the upstream control region. What are the transacting factors? We know from our Pol-2 example that if there are cis-acting elements, certain protein factors have to go and interact with the cis-acting elements. That is how the regulation takes place. In the case of Pol-2 transcription, it is the RNA polymerase II, general transcription factors and many transcription factors that act as transacting factors. In the case of Similarly, in the case of polymerase I also, there are transacting factors which interact with these two major cis-acting elements of rDNA promoter. One of the transacting factors or transcription factors involved in Pol-1 transcription is called as a core binding factor. This is the protein which interacts with the core region of the RNA polymerase I promoter. In the case of humans, for example, this core binding factor is known as selectivity factor or SL1. Selectivity factor 1 or SL1, whereas the same protein factor in the case of mouse is known as TIF1B, that is transcription initiation factor 1B. It is also various known as RAB1 etc. and so forth. Just remember, the human core binding factor is called as SL1, whereas the same thing in the mouse is called as TIF1B. Now, it is this SL1 factor which actually binds to the core sequence of the RNA polymerase I promoter. What is this SL1? What is the selectivity factor 1? SL1 actually contains the TADA binding protein and at least 3 TBP associated factors which are designated as TAF1 110, TAF 163, TAF 148. Remember, just like in the case of Pol-2 transcription also TADA binding protein is involved. In the case of Pol-1 transcription also TADA binding protein is involved, and among all the general transcription factors which I have studied, namely TF2D, B, E, A and F, it is the TADA binding protein which plays a very important role in the transcription of not only polymerase II but also by polymerase I as well as by RNA polymerase III. So TADA binding protein plays a very important role in the transcription of all three RNA polymerases. And just as the TADA binding protein in the case of Pol-2 transcription is associated with specific TAFs, we used to call as TAF2, 250 and so on and so forth. In the case of Pol-1 transcription also, there are specific TAFs, that is the TBP associated factors which are associated, but these are designated as TAF1. With the instead of TAF2 we call them as TAF1 depending upon the molecular weight. TAF1 110 means it is a 110 kilodalton protein, TAF1 63 means a 63 kilodalton protein, TAF1 48 means the 48 kilodalton protein. The TAFs which are required for Pol-1 transcription is different, the TAFs which are required for Pol-2 transcription is different. So, the TBT is required not only for Pol II but also Pol II transcription. However, the TAFs present in SL1 are different from those present in TF2D. Okay. So, the TBP in association with specific TAFs forms TF2D, and this TF2D is involved in Pol II transcription. But the same TBP in association with the TAF1 specific factors is called as SL1, and that is involved in Pol I transcription. SL1 cannot promote Pol II transcription, TF2D cannot promote Pol I transcription. These are distinct. So, TBP by interacting with either Pol I specific factors or Pol II specific factors can promote Pol II transcription or Pol I transcription respectively. Okay. The second important transacting factors are called as the upstream binding factors or UBF, which actually interact with the UCE region of the Pol I promoter. Now, UBF is a single polypeptide that binds to the GC rich region of the upstream control element of the UCE of the Pol I promoter. Interestingly, in addition to this core promoter and UCE, the spacer between the UCE and core promoter plays a very important role in Pol I transcription. Okay. The spacing can be changed by distances involving integral numbers of turns of DNA, but not by distances that induce half turns. It is very important because the factors that bind to the UC and the factors that bind to core 2E have to be on the same side of the helix, and then only Pol I transcription takes place. And if you now change the spacer by half turns, then these proteins binding to them cannot be on the same side of the helix, and therefore Pol I transcription cannot happen. So, this distance between the core UCE and core promoter is very important, and this ensures that proteins that bind to core region, proteins that bind to UBF are on the same side of the helix. Now, the recombinant mammalian SL1, which consists of TBP and the 3 TAFs, cannot support efficient promoter specific RNA Pol I transcription in an in vitro transcription system. So, just like we had TF2D, TBP alone could not sufficient to initiate activate dependent transcription in the case of Pol II transcription. In the case of Pol I transcription, TBP and TAFs alone are not enough to support basal transcription in a cell free transcription system. In addition to the TAFs and TBP, you also require another protein which is known as RRN3. This RRN3 is actually required for efficient formation of pre-initiation complex in the case of the Pol I promoter. This RRN3, which is actually known as TIF1A in the case of the mice, if you knock out this RRN3, the mice can survive only up to 9.5 days of embryonic development. After that they die. And even in these embryos, these embryos are very small because they contain very few cells. So, if you do not have RRN3, the embryos die after day 9.5, and the cells are also very small in these embryos, clearly indicating that in addition to TBP and these 3 TAFs which are Pol I specific, this RRN3 plays a very important role for efficient transcription of the Pol I rDNA promoter by RNA polymerase I. So, the SL1 in combination with RRN3 is required for the basal transcription, and these factors, the SL1, when it interacts with the UBF, which is binding to the upstream control region, now you get much higher levels of transcription. And in addition to this SL1, the TAF148 and TBP are also involved in the interaction between the UBF and the basal transcription in the case of RNA polymerase I. Now, so the core promoter region, the transcription factors which bind to the core promoter region consists of, is called as SL1 or selectivity factor 1. In the case of mouse, it is called as TIF1, it is called as TIF1B. So, the SL1 consists of Tata binding protein and TAF110, TAF163, TAF148. In addition to these 3, you require another protein factor called as RRN3 for basal transcription of RNA polymerase I promoter. This is about the core region or core promoter region of the RNA polymerase I. Now, if you move to the upstream region, the upstream control element binds to a protein called as UBF1. The UBF1 or the upstream binding factor 1 is a 97 kilodalton protein, consists of about 767 amino acids, and this is a cartoon that depicts the structure of the UBF1. It basically consists of 3 important regions. It contains what is called a homeodomain like region, which is involved in both DNA binding as well as dimerization. We have studied extensively what is homeodomain in our previous classes when we discussed about RNA polymerase II transcription, what kind of DNA binding motifs that transcription factors involved in RNA polymerase II transcription contain. zinc finger motifs, leucine zippers, helix loop helix, helix turn helix, homeodomain and so on and so forth. So, this transcription factor which is involved PAL1 also contains a region which is involved in DNA binding and dimerization, and it contains a homeodomain like region. Toward the C terminus, it contains a highly acidic region, that means it is rich in glutamic acid and aspartic acid, and this region is actually involved in interaction with the core binding factors, namely the SL1 factors. In addition, it contains a number of regions called as high mobility group boxes or HMG boxes. What are these HMG boxes? These HMG boxes share significant homology to what is called as a W80 amino acid domains on the nuclear non-histone HMG proteins HMG1 and HMG2, which are key structural elements in the chromatin and chromosomes. So, these regions have high mobility HMG boxes contain homology to proteins involved in chromatin structure and chromatin modulation. The UBF is actually involved. The reason why it contains this kind of HMG boxes is that the UBF actually plays a very important role in regulating RNA transcription in the chromatin context. In fact, the UBS is actually involved in formation of what is called as an enhanceosome, in which a dimer of UBS organizes 140 base pairs of the enhancer DNA into a 360-degree turn, as a result of which 6 in-phase bends are generated by 3 of the 6 HMG boxes in each UBF monomer. You can see what is the functional significance of the HMG boxes of the UBF factor. And formation of such structures at the upstream control element region under the core region would juxtapose these precisely spaced promoter elements, thereby supporting their interaction between SLF and UBF. So, these HMG boxes play a very important role in positioning these factors in such a way that they can interact, and the spacer also plays a very important role in this. Interaction of the UBF with the individual protein components of PAL1 machinery at sites other than the promoter throughout the RNA repeat might alter the chromatin structure and therefore enable access of such factors to the promoter and increase the local concentration of such factor, thereby indirectly enhancing the efficiency of pre-initiation complex formation. So, the UBF is just like the enhancer binding factors or transcription activators which we have studied in the case of PAL2 transcription. Now, very interestingly, a protein known as transcription termination factor or TTF1 binds to the termination sequence of upstream region of RNA promoter. As we seen in the case of PAL2 transcription, once transcription initiated, it also has to terminate. There are sequences in the case of PAL1 transcription which actually serve as terminator sites, and to these termination sequences specific proteins called as TTF1 actually binds and brings about termination of transcription. But one unique feature about this transcription terminator protein or the transcription termination factor 1 is that it plays a very important role not only in termination of PAL1 transcription, it also plays a very important role in the assembly of pre-initiation complex. It is a very rare example where a protein designation of a transcription termination factor actually promotes transcription initiation. How it does do it? It turns out the TTF1 actually interacts with a histone acetyl transferase, which we again studied extensively in our previous lectures, called P300 or Krebs binding protein, which actually has a histone acetyl transferase activity. So, you can see the hats are not only involved in the regulation of PAL2 transcription, but they are also involved in the regulation of PAL1 transcription. And we know this hat actually acylates one of the subunits of the core binding factors, namely the TAF168 of the SL1, and thereby enhancing the binding of the SL1 subunit to the RNA promoter. So, here we have a very interesting example where a transcription termination factor recruits a histone acetyl transferase, so that this histone acetyl transferase can actually acetylate one of the subunits involved in the core transcription factors and thereby enhance the rate of pre-initiation complex formation. And in addition, the TTF1 is also involved in what is called as remodeling of the chromatin, ribosomal chromatin, leading to eventual silencing of the rDNA gene. We will come to this little bit later during this talk. I would not like to confuse about the role of TTF1 in chromatin remodeling. We will discuss this particular aspect little bit later. Remember, the TTF1, which is involved in transcription termination in the case of RNA transcription, also plays a very important role in enhancing the rate of transcription initiation, because it recruits a histone acetyl transferase and makes it acetylate one of the core binding factors, namely TAF168 of the SL1. So, how does the transcription proceed in the case of RNA polymerase 1 or the RNA promoter? As we have seen in the case of the PAL2 promoter, The first step is the formation of pre-initiation complex, and this results in the initiation of transcription. Then you have promoter escape and promoter clearance. The initiation complex now starts elongation, ultimately transcription initiation, and then the polymerase 1 again has to come back, and with the help of initiation factors starts reinitiation, and that is how the polymerase 1 transcription cycle continues. So, let us now discuss what are the key steps which are actually involved in the RNA polymerase 1 transcription. The pre-initiation complex formation in the case of POL1 transcription involves the binding of the SL1 to the rDNA promoter, incorporation of the upstream binding protein, and recruitment of POL1B by SL1. So, these two factors, namely the SL1 and UBF1, when they go and bind to the respective regions, and recruitment of RNA polymerase 1 is one of the first step in the initiation of transcript by RNA polymerase 1. POL1 then initiate transcription, and following promoter escape, POL1 is converted into a process of enzyme called POL1 epsilon, which is involved in elongation of the nascent RNA. Just like in the case of RNA polymerase 2, the A is converted into O, or the non-phosphorylated form is converted into phosphorylated form. In the case of polymerase 1 also, it is converted to a specific complex polymerase epsilon, which is now involved in carries out the elongation of the nascent ribosomal RNA transcription. And once the POL1 escapes and clears the promoter, the UBF and SL1 remain bound to the promoter in vitro. Therefore, it can recruit one more molecule of POL1 and initiate another round of transcription. And in fact, it is because of this, that means the same UBF and S1 can recruit initiate multiple rounds of transcription, and therefore multiple RNA polymerases can start transcribing the rRNA gene. And finally, the transcription of RNA polymerase 1 terminates at the 3' end of the gene at specific sequences, and this is carried out by the transcription factor, termination factor TTF1, and also another factor called as PTRF, or POL1 and transcript release factor, and as a result of these interactions, the POL1 is released, and therefore it can go back again interact with UBF and SL1 and initiate one more round of transcription. This is how, in brief, the POL1 transcription cycle proceeds. Now, the important aspect that we have to discuss, just now we spent considerable amount of time discussing the mechanism by which POL2 transcription is regulated. Let us now try to understand how is ribosomal DNA transcription is regulated, how does RNA polymerase 1 activity is regulated inside the cells. This is very important, because I told you 50% of the total RNA inside the cells is accounted for by ribosomal RNA. Now, you do not require this amount of ribosomal RNA all the time inside the cells. So, only in actively growing cells you may have to require more protein synthesizing capability, therefore, you may require more amounts of rDNA transcription, whereas cells which are not metabolically active, you may not require that kind of a protein synthesis machinery, and therefore, it is a waste to have a continuous rDNA transcription. So, depending upon the physiological status, depending upon the metabolic status of the cell, the rDNA transcription is very finely regulated. Now, let us now take one or two examples and see how does the rDNA transcription is regulated inside the cells. Now, the rate of cell growth and proliferation is directly proportional to the rate of protein synthesis, which is intricately linked to the ribosome biogenesis, and therefore control the level of rDNA transcription by RNA polymerase 1. Now, if cells have to grow actively, they have to make lots of proteins, and if cells have to make lots of proteins, they require lots of ribosomes. If more ribosomes are required, then more ribosomal RNA transcription has to take place, and more of the messenger are involved in ribosomal protein subunits have to be made, right. So, if you have to have an actively protein synthesizing cell, you need to enhance the rate of rDNA transcription. So, depending upon what stage of cell cycle the cells are, the rDNA transcription also is very finely regulated. So, in addition to the requirement of the protein synthesizing capability, depending upon the cell cycle stage also, the rDNA transcription is very finely regulated. For example, transcription is completely absent. The rDNA transcription is completely absent during mitosis, but then gradually increases during G1 phase of cell cycle, is the maximum during S phase and G2 phase of the cell cycle. So, once the cell enter mitosis, the rDNA transcription is completely shut off. Why does this happen? That is because the nucleolus disintegrates. When cells enter mitosis, the nuclear membrane breaks down, there is no nucleolus, and therefore there is no rDNA transcription takes place. Since there is no nucleus, rDNA transcription is completely shut off. So, during mitosis there is no rDNA transcription, and once the mitosis is over and the nucleus reappears, again nucleolus is formed, and then rDNA transcription is starting. So, remember, this is a very important point. During mitosis, since there is no nuclear membrane, there is no nucleus and no nucleolus, RNA transcription is completely shut off during mitosis, and then gradually when the nucleus is reorganized and nucleus is reformed, RNA transcription resumes. And the phosphorylation status, how does this happen? The phosphorylation status of SL1 also fluctuates during cell cycle. Phosphorylation of the SL1 subunit of TAF1110 by cell site specific protein kinases during metaphase correlates with the inactivation of SL1. So, if we have to shut off RNA transcription, it turns out there are certain specific cell cycle specific kinases which go and phosphorylate the TAF110 subunit of the SL1, and as a result is inactivated. And therefore, SL can no longer interact with EBF, leading to mitotic repression of RNA transcription. So, during mitosis, a specific kinase called CDC2 cyclin B goes and phosphorylates the TAF110 subunit of the SL1 transcription factor, and when this is phosphorylated, it can no longer interact with the UBF transcription factor, and as a result POL1 transcription cannot take place. So, one of the mechanisms by which RNA transcription is regulated is by cell cycle specific phosphorylation of one of the subunits of the SL1. When it becomes phosphorylated, it is inactivated, and therefore during mitosis RNA transcription is shut off. Similarly, many other factors involved in POL1 transcription like TTF1, RRN3, UBF etc. are also regulated by phosphorylation during different stages of cell cycle, but I do not want to give too much details at this stage and confuse you. Remember, one of the major mechanisms by which RNA transcription is regulated is by phosphorylation of individual components of the transacting factors involved in RNA transcription, such as SL1, TTF1, RRN3, RBF. All these are subjected to phosphorylation, dephosphorylation by specific kinases, and it is by this phosphorylation, dephosphorylation RNA transcription is regulated during different stages of cell cycle. In addition to this mechanism, in addition to the regulation of RNA transcription by differential phosphorylation of transcription factors, another major mechanisms by which RNA transcription is regulated is by what is called as a epigenetic regulation of RNA transcription. Just as we have seen in the case of POL2 transcription, phosphorylation of transcription factors is one mechanism by which POL2 transcription is regulated, and in the previous lectures I gave an example of, for example, CREP, where I told you when you treat cells with cyclic AMP activates an enzyme called protein kinase A. Protein kinase A now goes and phosphorylates a transcription factor called as CREP, cyclic AMP response binding element protein, which then once it is phosphorylated, in the phosphorylated form CREP interacts with the histone acetyltransferase called CBP or P300, and this results in recruitment of RNA polymerase II and enhancement of transcription. This is one mechanism. The same case here also we just discussed that depending upon the phosphorylation status of SL1 and other transcription factors, the RNA transcription can be regulated. And we also studied, in addition to those kinds of mechanisms, histone modifications and DNA methylation can also regulate POL2 transcription, and we discussed this as epigenetic regulation of gene expression. The same way, POL1 transcription is also subject to the epigenetic regulation of gene expression, and histone phosphorylation or histone modifications as well as DNA methylation plays an important role not only in the regulation of POL2 transcription but also in the regulation of POL1 transcription. For example, the transcription termination factor TTF1 recruits an ATP dependent nuclear remodeling complex called NORC, and DNA methyl transferases called DNM1 and DNM3B as well as histone deacylase HDAC1 interact with this nuclear remodeling complex, and as a result it results in DNA methylation as well as histone deacylation. So, what is the mechanism by which RNA transcription is regulated epigenetically? It actually involves the transcription termination factor 1. It actually recruits a chromatin remodeling factor called NORC, nuclear remodeling complex, and this nuclear remodeling complex now interacts with certain specific DNA methyl transferases as well as a histone deacylase called HDAC1, and as a result the RNA promoter is methylated and the histones in the RNA promoter are deacylated, and this results in the prevention of binding of the UBF to the methylated promoter, and as a result RNA transcription is suppressed. So, phosphorylation of transcription factors involved in RNA transcription as well as DNA methylation and histone deacylation are two major mechanisms by which RNA transcription is shut off. But the epigenetic, this mechanism what we are discussing now is actually involved in what is called RNA silencing and is actually a long term RNA repression. Methylation of the histone H3 on lysine 9 and subsequent binding to of heterochromatin protein HP1 ultimately helps the condensation of nucleosomal DNA into silenced chromatin. So, rDNA silencing basically involves methylation of the H3 on the lysine 9 residue, and these methylated lysines are recognized by this HP1 protein, which again we discussed in detail when we discussed about histone phosphorylation, histone methylation in the previous classes. And once the HP1 bonds, it initiates heterochromatinization of the promoter region, leading to shut off of the transcription. And in fact, what is the evidence that the epigenetic regulation of transcription takes place in the case of POL1? If you actually treat cells which inhibits cytosine methylation as well as inhibits histodistillation, it actually can result in enhancement of RNA transcription, clearly indicating that cytosine methylation as well as histone distillation play a very important role in the regulation of RNA transcription, and this is the major mechanism by which RNA transcription is regulated in a epigenetic manner. Now, let us spend some time to understand how is the precursor rRNA is processed. Like I told you, a single 48S rRNA is synthesized from the ribosomal DNA, and then this is then cleaved into 28S, 18S and 5.S rRNAs. How does it take place? So, following their synthesis in the nucleolus, nascent pre-ribosomal RNAs are immediately bound by proteins to form pre-ribonucleoprotein complex, are called pre-rRNP. Again, you can see all these are analogous to what we discussed in the case of the POL2 transcription. Just as in the case of POL2 transcription, you have capping factors, you have splicing factors and polyadenylation factors, which all go and interact with the RNA and form RNPs or ribonucleoproteins. The same way, immediately after the synthesis of the ribosomal RNA, these ribosomal RNA also interact specifically with protein molecules, resulting in the formation of ribosomal ribonucleoproteins or rRNPs. So these proteins actually process these pre-rRNPs so that you get 28S, 18S and 5.S rRNA. And during this processing, pre-RNA undergoes several post-transcriptional modifications, just like the mRNA undergoes mRNA capping, the cytosine is modified a 5-methyl cytosine RNA in the 5' end. Similarly, the pre-RNA also undergoes several base modifications, such as methylation of the 2' hydroxyl group of specific ribose sugars, as well as conversion of specific uridine residues into pseudo-uridine residues. So, specific base modifications also take place in the case of the as a part of RNA processing in the case of pre-rRNA processing. The positions of the cleavage sites in pre-rRNA as well as the 2' O methylation pattern and pseudo-uridine formation are determined by at least 150 different small nucleolus restricted RNAs, and these are called as small nucleolar RNAs or SNO RNAs. So, just like we have all these UNSN RNPs which are involved in USN RNP in the case of splicing, similarly you have what are called as SNO RNAs which are actually involved in the processing of pre-rRNA to get mature ribosomal rRNAs. I will not go into the exact mechanism by which this kind of pre-rRNA processing takes place, it is a little bit advanced, so we do not have to worry about it. Just remember, pre-rRNA processing involves base modifications such as conversion of uridine to pseudo-uridines as well as modification of the ribose sugars. Also, it involves specific cleavage of the 48S rRNA precursor so that you get 28S, 18S and 5.8S RNA, and these entire reactions are catalyzed by specific RNA molecules known as small nucleolar RNAs, which are present uniquely inside the nucleolus. And these SNO RNAs are uniquely involved in the processing of pre-ribosomal RNA molecules. So, I will just put a cartoon here just to register in your mind what are the key steps involved in rRNA processing. For example, pre-RNP processing involves as soon as the 48S RNA is transcribed, it immediately associates with a number of proteins so that the rRNA now becomes a 80S pre-ribosomal ribonucleoprotein or pre-rRNP. 80S, about a molecular weight of 18S. So, the 80S pre-rRNP contains both 48S rRNA shown in red here as well as protein components, and then this USNO, the SNO RNAs or the small nuclear RNAs now come into picture and cleave this 48S RNA at specific regions so that you get what is called 32S rRNA and a 20S rRNA. And then this 32S rRNA is then processed to generate a 28S rRNA and a 5.S rRNA shown here in red, whereas the 20S rRNA is finally processed into 18S ribosomal RNA. So, through a series of steps, through the involvement of this SNO rRNAs, the 48S RNA is ultimately processed into 28S RNA, 5.S RNA and 8S RNA with 32S rRNA and 20S RNA as the intermediates. So, ultimately these 5.8S RNA, 80S rRNA and 28S rRNA form the components of the ribosomal subunits. The 18S rRNA, for example, is a component of the 40S ribosomal subunit. The 28S and 5.S RNA form the components of the 60S ribosomal subunit. In addition, the 5S rRNA, the synthesized by the polymerase 3, also joins and becomes a component of the ribosome. So, the RNA components of the ribosome, 3 of them come from ribosomal DNA and 1 of them come from the Pol 3 transcription. So, let us now spend some time to understand what is the significance of study of ribosomal DNA transcription. Why should we understand the regulation of RNA transcription? So far I discussed what are the major cis-acting elements involved in Pol 1 transcription, and we also discussed what are the transcription factors which bind to the core promote region as well as the upstream control region, and how RNA transcription is regulated in a cell cycle specific manner. And we know that during mitosis, because the nucleus and nucleolus disintegrate, there is no RNA transcription, and during this process, during mitosis, certain factors like the SL1 also undergoes, or other factors like UBF, they are also phosphorylated in a cell cycle specific manner, leading to their inactivation. So, phosphorylation, dephosphorylation of these transacting factor is one mechanism by which RNA transcription is regulated. And I also discussed what is called as RNA silencing, which involves epigenetic mechanism of regulation, wherein DNA methylation as well as histone deacetylation plays a very important role in heterochromatinizing the RNA transcription leading to RNA silencing. And then we discussed the RNA processing, how rRNA is processed into 28S, 18S and 5.S RNA, and a unique RNA molecules called small nucleolar RNA or SNO RNAs play a very important role in rRNA processing and generation of these 3 different rRNA molecules, which together with the 5S RNA synthesized by RNA polymerase 3 contribute to the RNA component of the ribosome. This is the major mechanism by which ribosomal RNA is synthesized and how RNA transcription is regulated. Now, let us see in the context of physiological context, what is the significance of study of ribosomal DNA transcription. The number of active rRNA genes varies between different cell types even within the same organism. Okay. So, even if you take the ribosomal DNA transcription from liver is different from that transcription in muscle, brain and so on and so forth, because it depends on the metabolic status of the cell type. Similarly, during cellular differentiation, for example spermatogenesis, there is a dramatic decrease in ribosomal RNA synthesis. In the contrast, when you stimulate cell proliferation, for example, if you take lymphocytes and add molecules like phytohemagglutinin (PHA), it enhances cell proliferation, and during such cell proliferation, there is a dramatic increase in the number of rRNA gene transcriptions as well as rRNA synthesis. So, when cells undergo differentiation, there seems to be a decrease in rRNA synthesis or RNA transcription, but when you stimulate cell proliferation, there seems to be an increase in the RNA transcription. Now, although there are hundreds and hundreds of copies of RNA genes inside the nucleus, not all these RNA genes are transcribed at a time. Only about 50% of the RNA genes are transcribed in each cell during log phase, whereas during stationary phase, the percentage of actively transcribed RNA genes is reduced owing to changes in the chromatin. So, depending upon whether cells are actively dividing or whether cells are in the resting phase or stationary phase, the percentage of RNA genes that are transcribed varies. And in fact, histone deacetylation mediated by RPD Syn3 histone deacetylase complex has been shown to be involved in the inactivation of RNA transcription, and as we discussed in the previous slides, this constitutes epigenetic regulation of RNA transcription. There are very interesting experiments have been conducted to actually study RNA transcription. For example, in the case of these cells which have been engineered to contain 40 copies of rRNA genes instead of the normal 75, and they asked the question, do they produce same level of RNA? That is, if you reduce the number of RNA copies, does the rRNA transcription also go down or does it remain the same? Very interestingly, they found although you decrease the number of copies of rRNA genes, they still grow at the same rate as the wild type yeast cells, and the amount of RNA produced still was the same. And it turns out that is why this kind of experiment actually told that not all the rDNA copies are transcribed at the same time. Depending upon the need, only a percentage of the rDNA genes are transcribed, and this can be finely regulated depending upon the need of the cells. Similarly, cell growth, when cells grow, there is actually an increase in size as well as increase in the mass, whereas when cells divide, there is an increase in the cell number. All these events are intricately linked, okay? And cell growth without cell division leads to production of large cells with a single nucleus, and in such cells there is a dramatic increase in the ribosomal RNA, ribosomes as well as protein synthesis. So, when you have huge cells with a huge amount of cell volume, there is a high levels of ribosomal DNA transcription, and the rDNA transcriptions also increase several fold in cancer cells, in actively dividing cells. The rRNA is produced at abnormally high levels in almost every tumor type investigated, indicating that overexpression of rRNA may be a general feature of all types of cancers. In fact, people are even thinking if you measure the rDNA transcription, it can serve as a prognostic marker for diagnosing cancer, because invariably in all cancer cells there is a dramatic increase in the rDNA transcription. So, the levels of rDNA transcription can serve as prognostic markers for cancer. And when cells are treated with chemicals that block rDNA transcription, it results in cell cycle arrest. I just given an example to actually show how Pol-1 transcription actually can be regulated in cancer cells. As we know, cancer is either because of overexpression of oncogenes or lack of tumor suppressor genes. So, just like an automobile has a accelerator and a brake, if oncogenes are overexpressed, that if you want the automobile to speed up and lose control, you press accelerator too fast, or if you do not want the automobile to stop, if the brake fails, then also it can result in the accident. The same way, cancer can be either because of overexpression of oncogenes or lack of expression of tumor suppressor genes, and people have actually found that the proteins such as c-Myc, RAS and RAF, which are actually oncoproteins, they all activate Pol-1 transcription, whereas transcription factors like RB and P53, which actually act as brakes, which inhibit cell proliferation, they actually inhibit Pol-1 transcription. So, there is a direct correlation between proteins which actually enhance cancer, which cause cancer, they activate Pol-1 transcription, or proteins which inhibit cell proliferation or act as tumor suppressor genes, they seem to inhibit rDNA transcription, indicating that there is a very good correlation between cancer phenotype and the level of rDNA transcription. There are very interesting experiments which have been done to actually visualize rDNA transcription. As early as 1969, for example, Miller and BT have actually demonstrated how you can actually demonstrate the actively transcribing rDNA genes, and you can actually demonstrate these are actually called as Christmas trees. So, the molecular organization of active ribosomal gene in the form of Christmas trees was described by Miller and collaborators in nucleolar spreads from amphibian oocytes more than 40 years ago. In fact, these are popularly known as Miller spreads. So, you can actually see actively transcribing RNA genes, and you can actually demonstrate RNA is actually coming out. These are actually called as Christmas trees. So, what I have actually done in this particular class is to describe in detail the mechanism by which the organization of ribosomal DNA genes, the mechanism by which ribosomal DNA genes are transcribed, as well as the regulation of ribosomal DNA transcription. And I made it very important to made it a point to emphasize the fact that ribosomal DNA transcription plays a very important role, because unless there are ribosomes, cells cannot make proteins. Therefore, depending upon if the cells have to make large amount of proteins, they have to need large amounts of ribosomes. That means the rDNA transcription has to go on very high levels, and depending upon the physiological status of the cell, metabolic status of the cell, and depending upon the cell cycles, the rDNA transcription is very finely regulated. There are two major mechanisms of regulation of rDNA transcription. One is post-translational modifications of transcription factors like UBF, SL1 and so on and so forth. Other major mechanism is called as rDNA silencing, which involves DNA methylation or methylation of the rDNA promoters, as well as histone deacetylation leading to heterochromatinization, and this is what is called as an rDNA silencing. I think with this I have completed both Pol-2 transcription and Pol-1 transcription, and the next class we will study how Pol-3 transcription is regulated and what is the significance, what is the mechanism by which tRNA genes are transcribed and tRNA synthesis is regulated inside the cells. I think I will stop here.